Hi, my name is Jian Zhang from the University of Queensland. I'll be introducing how to run SBayesR in this video. I will start with introducing the core features of SBayesR. SBayesR is a method based on the BayesR model with a focus on improving the polygenic score prediction for complex traits. The BayesR method was originally developed for predicting breeding values in livestock genetics using individual level genotype and phenotype data. Recently, we extended the BayesR methods such that it only requires GWAS summary statistics and LD matrix from a reference, and we called it SBayesR. So similar to BayesR, it can be flexible to account for a variety of genetic architecture of complex traits. SBayesR make use of publicly available GWAS data with a very large sample size, and it is more computationally efficient. We can quickly derive the summary data-based model as below. So let's consider an individual data model with a standardized genotype matrix X. If we multiply both sides by 1 over n times x prime, it gives this new equation. It is recognized that the left-hand side of this equation is the GWAS marginal SNP effect estimates. And on the right-hand side, we have the LD correlation matrix times the SNP joint effect and plus residuals. So this is a general form of model underlying many summary data-based methods. In SBayesR, each SNP effect is assumed to have a mixture prior distribution of 4 components. Specifically, it includes a point mass at 0 and 3 normal distribution with different variances. The variances are scaled such as it represents the small, medium, and large effects. The common factor of the variances in the normal distribution is assumed to be unknown and with the prior distribution. The mixing probability are also assumed to be unknown and to be estimated from the data. So in SBayesR, the SNP effect can be written as the weighted sum of 4 distributions. And the benefit of this prior is that it can nearly well approximate all forms of distribution for the true SNP effect, even with those distributions that are not normally distributed. This is why we say SBayesR is flexible in accounting for different genetic architectures. In SBayesR, we estimate the SNP effect by MCMC sampling, and the SNP effect are estimated by the posterior mean of the posterior distribution. Then we can compute the polygenic score by using the joint effect of SNPs across the whole genome. The SBayesR method is implemented in GCTP software, and this is our website. In the download page, we provide the executable file compiled on the Linux platform with the source code. And we also provide the LD matrices that are used in our paper. And we also provide tutorial data that include the scripts and example data to run our software. On our website, we also have a tutorial page that includes the key steps that can walk you through how to use our software and using the example data set. So the minimum command of using running SBayesR include the following components. The first component is a flag to specify the method, because our software GCTP include not only the SBayesR method, but other Bayesian methods as well. This flag specifies the input file for the GWAS summary statistics. And we also need a flag to input the LD matrix. --out specify the output file name for all the output files. And this is the part where we direct both the console output and error message into the log file. because it's usually quite useful to check the intermediate results in your log file. And you can specify other settings, including the model parameters and MCMC parameters. And we'll come back to that later. For now, the minimum command just to use the default settings for these parameters. So next, I first introduce how do we prepare the GWAS summary data file. The GCTB uses the same summary data file format as in GCTA COJO analysis, which is the text format file, including the SNP names, A1, A2 allele frequency for A1 allele, GWAS effect size for A1 allele, the standard error of the GWAS effect size, p-value, and the per SNP sample size. It is important to note that the GWAS effect size are at the A1 allele, which is the effective allele, which is the effect allele. And the transformation of your GWAS data to this .mar format would depend on the software you use to run GWAS. In our GCTB tutorial data folder, we provide a simple R script to convert the p-link linear model output to our .mar file. If you are using a GWAS from case control study, then you need to use the log odds ratio as the beta value or the B value in the MAR file. Next, we show how to prepare the LD matrix for our analysis. If your GWAS summary statistics are from European samples, then you can just use the LD matrices provided by our website. And if you want to compute the LD matrix by yourself, it is very important to remember that using a reference population that is as close as possible to the GWAS cohort, at least in the ancestry genetic information, is quite important. Otherwise, if there is a systematic difference in LD between GWAS and reference sample, it may cause a convergence problem. This command computes the full chromosome-wise LD matrix using the p-link BAT file as the input genotype file. It will produce a binary file, which includes all the matrix elements stored in the float position, and also an information file, including the SNP information, as well as the window information. And this is shows the number of SNPs that are in LD with the SNP. Because we are computing the chromosome-wise full LD matrix, so this window contains as many SNPs as on the chromosome. When we compute the sparse or the shrunk or the banded matrix, you may see a variability in the window size across SNPs. Depending on the number of SNPs and the sample size, computing the LD matrix for the entire chromosome can be quite memory and time consuming. So to improve the computation efficiency, we provide an option to partition the LD matrix into n parts and compute each part in parallel. So this flag --part m, I specify the run for i's part out of the total of n parts. This could be particularly useful when you are running a job array on HPC cluster. This command will generate the LD data file for each part and also a text file which contains the file name for each part. And then we run this command with the generated text file to merge all the parts together into a single file once all the parts are completed. Using the full chromosome-wise matrix for the genome-wide analysis can be quite memory consuming. It may take over 200 gigabytes. So to reduce the computation burden, in practice, we used either a shrunk matrix which shrinking the off-diagonal values towards 0, or a banded LD matrix, which only include the LD correlations in a fixed bandwidth for each SNP. And this plot shows the shrunk LD matrix. The banded matrix would be similar, but may have a wider bandwidth. So next, I'll show how we compute the shrunk matrix. It will be similar for computing the banded matrix, and the information can be found on our website. So the algorithm for computing the shrunk matrix is provided by Wen and Stevens' paper in 2010. The calculation of the shrunk LD matrix would require a genetic map that includes the genetic position for each SNP. So one option to compute the shrunk matrix is to read in the full LD matrix that we have computed previously and make it shrunk with this provided the genetic map. And in the public domain, there is a genetic map for European population available to download. And then this command will generate the LD matrix in a sparse format, which is more memory efficient. And if you were to use other genetic map, you might need to specify the effective population size and the genetic map sample size to get appropriate shrinkage parameter. Another way to compute the shrunk LD matrix is to directly compute it from the genotype file, and it can also be computed by parts. Once we have the shrunk LD matrix for each chromosome, the last step is to produce a text format file that includes the path and name of each chromosome by rows. And then we can give this text file to this command to compute to perform the SBayesR analysis. And just know that the LD matrices from HAMAP3 SNPs or more common imputed SNPs in European samples are available to download from our website. And in practice, we found the shrunk LD matrix are robust in general and more computationally efficient. But for some traits, using the banded matrix with 3 centimorgan bandwidth, as suggested by the authors of LD-PRA2, can lead to a higher prediction performance for some traits. And here it shows the specification of some model parameters and MCMC settings. The -- pi follows a number of decimal numbers separated by comma. And these numbers, the number of these decimal values specify the number of the mixture components in the model. And these numbers specify the starting value of each mixture component. If you only specify 2 components, that will reduce to a SBayesC model or a LD-PRA-like model. And this gamma value specify the weights for the variance component. And the number of gamma values should be consistent with the number of values for pi. Chain length specify the total number of iterations for MCMC sampling, and burn-in is the number of initial iterations to be discarded when collecting the posterior samples. And here shows a part of the log file for outputting the intermediate result. It's always good to check the intermediate result. Sometimes it can flag an issue. For example, here shows the phenotypic variance estimated from the SNP summary statistics. And here shows the GWAS SNP sample size, the mean and the standard deviation. If the standard deviation is too large, you may want to consider remove some outliers. And here shows for every 100 iteration, what's the number of SNPs fitted in each of the 4 mixture components, and what's the sample value for residual and genetic variance and SNP-based heritability. And the last column shows the predicted time to finish. And once the job is finished, it generates a parameter result output file. which shows the posterior mean and the standard deviation for the key parameters. We use the posterior mean as the point estimator and the standard deviation from MCMC samples as the posterior standard error estimator. Another important output file is the SNP result. Here shows the SNP information in this file and also the posterior mean of the SNP effect, posterior standard error of the SNP effect, and the posterior inclusion probability. This is the Bayesian measure of the trait association. Higher the BIP value, the stronger the evidence for the association of the SNP on the trait. The last column shows the sampled value of the SNP effect in the final iteration. And then we can calculate the polygenic score using the P-Link software by input the validation genotype data using the bad file flag and using the score flag to input the SBayesR estimated SNP effect. Only the second, fifth, and eighth column are needed. Finally, I would like to briefly touch on a convergence issue when you run the SBayesR analysis. This could happen sometimes, especially when the summary statistics from the meta-analysis are used. When it happens, the effect size are blowing up and the program may be terminated by reporting this error message. And this could happen when there is errors in summary statistics, such as errors in allele frequency or per SNP sample size. It could also happen when there is a systematic difference in LD between GWAS and reference samples. We suggested to first check the distribution of the per SNP sample size, if such information are available, and remove the outliers. For example, in this plot, there is a long tail on the left, and having these SNPs in the model might be detrimental because that would misspecify the residual variance covariance structure. If the per SNP sample size information is not available, you can use the impute n function to impute per SNP sample size based on other summary statistics, and the program will do the filtering for you. We also find that during the MCMC sample, if we update the per SNP, the SNP size variance as a function of the total genetic variance and the pi sample values, that would lead to a more robust performance. And this can be specified by the robust flag. In fact, the program will switch to this more robust parameterization mode once it detects a convergence issue. And more suggestions, and please check our FAQ page on our website. And at the end, I would like to thank my supervisors and the co-authors of the paper and the funding source. And thank you very much.